Hello my dear students, in today's class we will unlock the fascinating process of urine formation and the countercurrent mechanism, two key pillars of renal physiology. Did you know your kidneys filter nearly 180 liters of fluid every single day, yet you don't lose all that water thanks to the brilliant design that helps conserve water, salts and vital nutrients. So how do these bean-shaped organs pull off such smart and selective process? Get ready to unlock the secrets of the nephron, understand how urine is formed and discover the amazing countercurrent system that keeps your body balanced. Let's begin this exciting journey through the microscopic marvels of your kidneys. Urine formation is the kidney's way of filtering and cleansing the blood. About 1300 ml of blood, around 26% of cardiac output, flows through the kidneys every minute, producing 1 to 1.5 liters of urine daily. This process involves three main steps: glomerular filtration, where blood plasma is filtered through the walls of glomerular capillaries into the Bowman's capsule. Water and small solutes pass through, forming the initial filtrate. Tubular reabsorption. As the filtrate moves through the renal tubules, about 99% of water and essential substances like glucose, amino acids and ions are reabsorbed into the bloodstream via peritubular capillaries and the Vasa Recta. Third one is the tubular secretion. Waste products, excess ions and drugs are secreted from the blood into the tubules, helping fine-tune the urine composition. Together, these steps maintain the body's fluid balance and remove waste efficiently. Glomerular filtration, the first step of urine formation, where blood is filtered from the glomerular capillaries into the Bowman's capsule through a three-layered membrane, endothelium of capillaries with tiny pores or fenestra, base membrane acting as a barrier, podocytes of Bowman's capsule forming filtration slits. Plasma, except large proteins, passes through, forming the glomerular filtrate, a mix of water, salts, glucose and waste. This process is known as ultrafiltration, and the glomerular filtration rate GFR is about 125 milliliter per minute or 180 liter per day in a healthy adult. Factors affecting glomerular filtration rate. Glomerular filtration rate or GFR is influenced by several key factors: increased renal blood flow, that is approximately 1300 ml per minute, and glomerular pressure, that is approximately 60 mmHg, enhanced GFR, while high colloidal pressure nearly 25 mmHg and back pressure in Bowman's capsule reduce it. Macula densa senses salt levels and adjust GFR via feedback. Narrowing of afferent arterioles lowers GFR, whereas efferent arterial constriction may first raise and then lower it. GFR remains stable between 60 to 180 mmHg arterial pressure due to autoregulation. Strong sympathetic activity can reduce GFR, as can reduced capillary surface area, increased capillary permeability or mesangial cell contractions. Hormones like atrial natriuretic peptide, BNP, NO, dopamine or prostaglandin E2 increase GFR or glomerular filtration rate, while angiotensin II, endothelins, noradrenaline and PGF2 decrease it. Now we'll study the second step of urine formation, that is tubular reabsorption. Tubular reabsorption is the process where the water and useful substances from the filtrate are taken back into the blood. As the filtrate flows through the nephron tubules, about 99% of water along with electrolytes, glucose, amino acids and other essential molecules are reabsorbed into the peritubular capillaries via the renal medulla's interstitial fluid. There are two main types of reabsorption. Active reabsorption requires energy to move substrate against a gradient. Examples: sodium, potassium, calcium, phosphate, glucose, amino acids, etc. And the second one is the passive reabsorption, Where no energy is needed, substances move along the gradient. Examples: chloride, urea and water. Reabsorption occurs throughout the entire nephron tubule, helping the body retain needed substances and maintain fluid and electrolyte balance. Tubular secretion, the third and final step of urine formation. This process moves substances like potassium, ammonia, hydrogen ions and urea from blood into the renal tubules to maintain chemical balance. Sites of secretion: potassium plus is secreted in the proximal and distal convoluted tubule and the collecting duct. Ammonia is secreted in the proximal convoluted tubule. H plus ion is secreted mostly in proximal convoluted tubule and also in some amount in distal convoluted tubule. Urea secretion occurs in loop of Henle. So urine formation and concentration, that is, out of 180 liter per day of filtrate, only 1 to 1.5 liter becomes urine. Filtrate osmolarity is 300 milliosmol per liter, but urine osmolarity up to 1200 milliosmol per liter. This process depends on body water level and ADH hormone, that is antidiuretic hormone. Concentration of urine occurs after filtrate reaches the distal tubule. Now we should know the production of dilute and concentrated urine. When the body has excess water, ADH secretion is suppressed, reducing water reabsorption in the kidneys and leading to the formation of dilute urine. In contrast, when the body is low on water, the kidneys conserve it by forming concentrated urine, which involves two key processes: the countercurrent mechanism that creates and maintains a medullary osmotic gradient, and the secretion of ADH, which increases water reabsorption from the renal tubules. This is the diagram showing the formation of dilute and concentrated urine. Countercurrent mechanism, it's a system is formed by U-shaped tubules where fluid flows in opposite directions in two limbs. Two main divisions of countercurrent mechanisms: one is countercurrent multiplier, which is found in loop of Henle, and the second one is countercurrent exchanger, which is located in Vasa Recta. The first one, that is countercurrent multiplier, there is loop of Henle. This is the diagram showing it is found mainly in juxtamedullary nephrons. The long loops reaching deep into the medulla and creates a medullary osmotic gradient to make medullary interstitial fluid hyperosmotic. But how this countercurrent multiplier works? Ascending limb actively reabsorbs Na plus and Cl minus and added to medullary interstitium and increases ultimately the osmolarity. Descending limb, it is permeable to water and absorbs Na plus and Cl minus passively from the medulla. Ions recirculate between ascending and descending limbs via the medullary interstitium. This recycling and new ion addition multiply the osmolarity of medulla, called a multiplier effect. Now we will study countercurrent exchanger, which is located in Vasa Recta. Vasa Recta preserves the medullary osmotic gradient set by the loop of Henle. It allows exchange of solutes and water without washing away the gradient. Role of antidiuretic hormone: without ADH, distal convoluted tubule and collecting duct are impermeable to water and it will form the dilute urine. With ADH they become permeable, so water is reabsorbed, called as facultative reabsorption. Final urine becomes hypertonic up to 212 milliosmol per liter due to water removal. Thank you so much for joining today's session on urine formation and the countercurrent mechanism. I hope this helped you understand how our kidneys work so efficiently to conserve water and maintain balance in our body. If you enjoyed this simplified approach to renal physiology, don't forget to like, share and subscribe. Stay curious, stay consistent, and I will see you in the next class. Bye.